Thank you so much for the invitation. It's a pleasure to be here. Thanks, Nusha, for a great talk. And thanks, Michelle, for carrying and everyone else for carrying the MRM. I think it's been great to see it take on and the community building around it. So I'm really happy that I can present here. And so maybe I think we'll have a bit of more technology-driven presentation than maybe what you're used to. So it's like Molly just introduced. Our lab, there's a lot of technology. But of course, being in biomedical engineering, we try to think about what could be the applications, and we're both thinking about patient care, but also about research tools. And so I'll jump right into kind of the outline. I mean, feel free to post questions in the chat. I'm not sure if I can see it actually. If there's something, I can try to answer. Also, maybe I think I'm supposed to answer at the end. So So we've kind of three multiplex techniques we're looking at, one on single cells, which Nusha just nicely introduced. Then we have had a big effort on extracellular vesicle analysis, so EVs, as I've abbreviated them here, which I will focus on. And then finally on multiplex affinity proteomics. And I was actually considering starting on that first, but seeing as time is moving forward, I put that in the end, and if time permits, I'll talk about it, or then maybe we can move more to the questions. And so I hope there won't be too many people disappointed about that. And so we're still in the early stage of these technologies, but I hope that this will kind of inspire you and maybe give you some ideas of how we might use them or how they might apply to some of your research as well. And it's always interesting to find potentially also collaborators and important applications for them. I have a kind of a generic thank you slides here. So we have had funding over the years from many different sources, as well as lots of very fruitful collaborations. And I will highlight a little bit more the ones that have been involved in this particular project. And of course, acknowledgements also go to the lab. And then here is a COVID kind of picture, which I think we've all had our challenges, but people have been really fantastic. And we're a wet lab, so we'll have the opportunity or the obligation as well, to some degree, to come back. And people have been very carrying through this pandemic. And so I want to thank them, all the great team that we have had in the lab. And I'll thank more specifically people involved in the project as well then. And so we're a technology development lab. I'm not going to dwell too long on it, but if you want to know more, you can go to our webpage. And they will explain the different things, and this is evolving as we focus on new projects. But by and large, our webpage is also fairly up to date. So one way of thinking about our technologies, how to contextualize them, was trying to apply them to better understand human health. And what we have been seeing over the last few years is how omics technologies, in particular, have really transformed our understanding of biology and our concept of how human health can be. And I speak about omics, but we also see now mobile health technologies and cell phone technologies coming up. And so there's been quite a transformation in terms of how we even conceive about the understanding of the human body. And so from the lab's perspective, we have a few areas where we are interested or have been contributing, and they're highlighted in green here. And you can think of them as human health application, but in many cases, they're also research tools for being used in the lab. So we can use them for liquid biopsies, like for CTCs, of course, which is the rare cells here, but we can also use them for cell supernatant or for cell extract analysis, or for CTC. It could also be for stem cell analysis, for example. And so the three are antibody-based proteomics, the clamp, so that's the bead-based assay that I'll tell about in the end, and the extracellular vesicle analysis using both microarrays and DNA exchange imaging that we have seen here for cell, but actually that we started developing initially on extracellular vesicles. And so EV analysis has been kind of a large construction project. I feel like our pace moves almost at the same pace as McGill construction projects, which take a long time in planning and a long time in execution. And it's because we dig quite deep, so we're building a strong foundation, and we have had a great team of working on that. And so they have been actually moving forward. As you will see, the results are here. But actually, we haven't published them, so most of what I will show is really unpublished work, or actually all of it. And so we hope to have it out soon. And we have also benefited from collaborators here with Janusz Irak, Peter Mitrakis, and Peter Siegel, since a lot of it was actually developed in the context of cancer. And so small extracellular vesicles, so what are these? These are lipid bilayer coated bubbles, 50 or 200 nanometer in size, and they have a bunch of cargo which we can sync as a potential biomarker or signature, and by carrying multiple of them together, they carry a signature of the cell of origin, which positions them in a unique way compared to traditional markers such as DNA or proteins, which are kind of isolated from their context. Now, when we're analyzing, what we're seeing is small EVs, and small EVs have actually really two distinct sources. One is through these multivesicular bodies, which is what has become as exosomes, and there's quite an excitement about them. But small EVs also comprise microvesicles, which are released directly from the cell membrane. And when we measure them, it's very difficult actually to know which source they're coming from. And so we're basically analyzing a mixture of these two vesicles. And that's one challenge that basically affects the field, because both of them express similar sets of proteins, but they can reflect different changes in terms of disease. And so what we look at is small EVs that we actually extract using size exclusion chromatography, and that we have characterized in terms of integrity and so on. So I'm not talking much, I won't talk much about more of the characterization or this initial characterization. And so EVs, our initial analysis platform was using our antibody microarray, which we developed initially for proteins. And so this is work that Rosalie started in the lab. So we're starting by spotting capture antibodies on a surface. And these antibodies are targeted here against tetraspanin, which are canonical proteins expressed in EVs that everyone uses to capture and characterize them. Then we incubate with the EVs. They will bind based on their receptor to different capture sites, knowing that each of them could potentially bind to any one of these capture sites here. And then we'll incubate with a detection. And in this case, we have to have one detection per well, because of course many different antibodies could bind on a single EVs. And then finally, we have fluorescent label, and so we can then scan in a microarray scanner to find out what exactly bound on each of these spots. And so the fluorescence intensity of each of these spots is then both a reflection of the number of EVs we bound and whether they express a particular protein that is being used for the detection. And so we can use the same antibody that we use for capture for detection, and that makes it pretty clear. But with EVs, we have the opportunity to mix and match. So we have a different capture antibody and the different detection antibodies. And as you can quickly imagine, this gives rise to a lot of different combinations that we can look at. And so this is the kind of images that we're getting and that we can then use for the analyzer. So here's a 1 millimeter scale bar, and the spots are about 100 to 250, 200 micrometer in size. And so one of the first things or an interesting sample we were looking at was a sample of metastasis. So this has a clinical motivation. For cancer and many other cancers, it's not the primary tumor that kills the patient, it's the metastasis. And actually, diseases metastasize in very specific ways to different organs. And so why is this metastatic happening to particular organs is one of the big questions that could influence both diagnosis and disease. And so working with Peter Siegel, he has developed a technique to extract cells that metastasize specifically to a particular organ. So organotropic, so-called organotropic cell lines. And the way this is done, they have an injection of an aggressive breast cancer tumor cell into a mice, where then the tumor colonizes the mice, and in this case the lungs. It's a breast cancer cell that goes to the lung, that grows there, and after this, the mouse is sacrificed, the cells extracted and are recultured and are re-injected into mice. And so this is an artificial selection process, and after several rounds of selection that way, we have actually cells that very strongly target the lung, because that's how we have selected them. And so you have an organotropic cell line. And we can then do the same for lung, for brain, for bone, and for different organs, and collect different cells with different organotropic potentials. And so for this metastatic cancer, we have then several ones. One is bone, so 1833, lung 4175, and then 2 brain metastatic cell lines. And then Peter Siegel, he actually had analyzed gene expression patterns, changes in these cells, but we can then expect that this might also give rise to changes in extracellular vesicle excretion in these cells as they change their organotropism. And there's indeed one seminal paper that showed that actually the secretion of EVs could prepare the metastatic site, the niche, so to speak, for colonization, for subsequent colonization by cancer cell. And so this has created a strong interest in trying to understand these mechanisms. And one of these molecules that actually modulate this binding of the cells to the metastatic site are integrins. So integrins are cell membrane, and they're pairwise combination, which is shown here. So beta-1 is one of these integrins that can be paired with many of these alpha integrin, which again can be paired with other beta integrins. And they can then bind, for example, RGD receptors or laminin receptors or collagen receptors. And these particular combination can fine-tune where a cell binds. And so we can now, for example, try to see how EVs from particular cells might express these integrins and how organotropism could change how these integrins are expressed within different extracellular vesicles. And so here are some preliminary results where we basically have EVs. We capture them with TIM4. So this is a protein that targets phosphatidylserine, which is expressed in all cancer extracellular vesicles. And so we can then look at the parental cell, and then at the bone, lung, and the two different brain cells, and see how the changes in expression will could affect or how the organotropism could change the expression of various proteins. And the most prominent one here are the tetraspanins, CD9 here. I think it's the most abundant one, but then also different integrins. Now, one challenge we're having here is that, of course, the concentration of EVs might change, and so this is always a question in terms of, are we capturing more EVs, but they have the same expression volume, or is it a smaller number of EVs with high expression of these integrins? And so, to some extent, this is mixed, but of course, what we can use here, since TIM4, we can expect roughly the same number of EVs, and so that this actually reflects true changes in integrin co-expression. We can then take this one in another way around, where we're looking now at the relative expression of these EVs that are being captured here with different proteins. But when we look at the overall expression of the EVs, not using a detection antibody, but using a fluorescence dye. And this is another way where now we can try to evaluate and quantify in a different way whether this EV expression might be real or whether there might be some confounding factor. And here we have basically the same graph shown on both sides with different expression levels, again, for these different cell lines. And so if we move this further and now to integrins, and here's a graph, and I don't have more detailed analysis of the meaning of the biological implications here. These are still preliminary results, but we can see that basically overall we have beta-1 that is overexpressed, and then we have typically here beta-3 that follows, and then different expression levels of the various different integrins. And so one of the questions that we had, I mean, this is kind of consistent, that you would have some enrichment of integrins, and we actually expect that the enrichment of a particular co-enrichment of two proteins would be actually significant for a biological modulation of the pathways. And so we expect this to be the case, and actually the previous work was also work by Molly, and but then how could we quantify whether something is truly enriched or co-enriched? And whether it's just basically because of the abundance. Let's say you have a capture antibody, you have a high abundance of those, and maybe you have a high abundance of different proteins. And so here we using our multi-labeling procedure, we introduce basically a normalization protocol that we can evaluate both the abundance and the expression level of the relative expression level of different antibodies. And so the first step was to evaluate the relative expression of a protein by first capturing it in a universal way with this TIM4 surface binder, then recording both the protein of the detection antibody, so the anti-B protein here, and together with the detection of this universal dye that is also including in the EV. And then we can simply calculate the ratio of these two values to evaluate the relative expression of this protein for this particular EV. And so this is using a universal capture. Now, taking this one step further, we can then also have a specific capture antibody and then try to evaluate whether, for example, one protein might enrich with this CD9. And we can then capture both, and by comparing the signal both for the universal capture and the specific capture, and having again the CFDAE as a normalization here, we can then compute the overall enrichment factor of a particular protein. And so the steps are basically captures EV on the anti-A capture antibody, again recording the fluorescence of the anti-B detection antibody and the CFDAE, and then we can capture a ratio of these two for a particular capture. So we have a ratio of the normal signal versus the other one, and then finally we can calculate an enrichment by dividing the ratio for the normal ratio, the ratio in the overall total population, divided by the ratio that we see in this enriched fraction. And so if this ratio is not the same, if this ratio is the same, that exactly the same ratio, that means we have no enrichment. So it's a random distribution of this molecule within the EVs. But if, let's say, there's a biological association, these two things are always seen together, then we would be able to see an enrichment of that molecule. Or conversely, if they are expressed through different biological pathways and don't mix together, then we will start to see a negative enrichment into these EVs. And so this allows us now to do enrichment analysis without actually needing access to single EV expression levels. Because, of course, if you can measure single EVs and co-express different EVs, then you could measure whether they're co-enriched. But here we can measure it directly or using, it's kind of an association pathway, using a binary association with a normalization step. And so this is then something that we have started looking at, this integrin pathway. And so we're looking at this integrin beta-1, alpha-6, or beta-1 alpha-6 or alpha-6 beta-4 as your detection, and looking at different subpopulations that are being captured. And we can see, and unfortunately, one thing we didn't do is actually the co-enrichment between these two, which is really what we should be doing. But we can see that alpha-6, which is prominent, we saw previously, is enriched here, and beta-4 is actually also enriched in quite a few of these populations here. And then we can repeat the same experiment with lung, with bone, and with brain. And we do see some differences here. Well, we generally see that beta-4, for example, is not really co-enriched in these CD ones, but is more enriched in the tetraspanin, but is more enriched in these other markers down here. And so broadly, what we see is alpha-6 co-enrichment in most organotropic EV subpopulations here with all these capture markers, and the beta-4 co-enrichment with brain organotropic EV populations in particular, which is what we're seeing down here, which is slightly different from here. And so we start to see some differences in these EVs here between these organotropic cells that could be contribute then to their organ targeting, because the integrins are involved in how they go to different organs. Then we here are the same basic type of measurement, but here we're looking at, instead of these integrins, we're now looking at different targets, so the CD63 and CD89, CD9, and EGFR. And we actually see that EGFR here, for example, is depleted in the parental cell line, whereas it's being more enriched in the organotropic cells, which to some degree, since EGFR is a epithelial growth factor receptor, is associated also with metastasis and with aggressiveness. We can see here that this is being overregulated in the organotropic cells and could be a reflection of the more aggressiveness of these cell lines. So these are preliminary results, and we still hope to extend them further and pursue further measurement on them. And so tetraspanins are canonical EV markers, and many people are using them. And so one of the questions is, are tetraspanin universally found in EVs? And this is most important because, and this is actually quite important, because we use tetraspanin often to decide whether something is an EV or not, especially, for example, in flow cytometry, where they use it. Does it have a tetraspanin? And if yes, then does it co-express something? Or then sometimes tetraspanins are used to enrich particular EV population. And in our result, what actually was quite striking is that we had several subpopulations where we found that the expression of these tetraspanin was actually could not be found, whereas we found other cancer markers, but so tetraspanins cannot necessarily be found in all the EV populations. And so, this is something that needs to be considered, and that shows that there's actually that we need to be very careful when we speak about EVs, and understand that actually no protein is actually expressed in all EVs. And so so now with these methods we can start to actually extract and evaluate these co-expression patterns. So this shows, and what we can do at the same time, of course, is then correlate the relative expression levels of these different proteins So EGFR, you can see, and HER2 here, they're being strongly enriched here. Also, the overall expression level actually a factor 10 in terms of how much they're expressed. And so that's one thing we need to remind when we look at co-expression. We don't have any idea here about the abundance. It only tells us whether something is statistically more highly expressed than what would be expected just by random distribution. Okay, and then in terms of applications, so we can also now use these tools for looking, for example, at changes in expression between 2D and 3D. So these are HEK cells, and Molly actually cultured them in 2D and 3D cultures. And here again, we're looking at EV subpopulations and looking at integrin alpha-1 expression over a time course. And we can just see in this case that we see different time courses, for example, here in 2D and in 3D, where integrin alpha-1 seems to be expressed more in 2D, but not so much in 3D, whereas for integrin beta-5, we can see not much expression in 2D, but then more expression in 3D. And showcasing again that we can see differences, that this might be a way to analyze difference, and that the multiplexing gives us a better view of all these different parameters. Another application that we're now exploring was to use organoids, or spheroids in this case, and we're looking at the secretion of EVs, and so it ties in our previous work. These are organotropic slides, and looking at the secretion and seeing how these different EVs over time might co-express different proteins. And so this is kind of a combination of the preliminary work in a particular context, and so that we can then try to evaluate how EV signaling might respond to the interaction between the cancer cell and the stromal cell within an in vitro metastatic niche on a chip, so to speak. Now, what I've shown you so far were all EVs that are measured on the surface of proteins, but a lot of cargo of these EVs is also inside of the EV, and so that's one of the points we wanted to try. Would it be possible to also measure the EVs inside of the proteins inside of this EV cargo? And Rosalie is the one who worked on this for now several years, and one of the things when we measure them, we can actually measure EV without cross-linking, but of course in many cases we do cross-link and we do permeabilize the EVs, and this would actually allow us to measure directly the proteins inside of these EV. However, following fixation, a lot of the proteins are not accessible. And so when we started doing that, this was not a very successful approach. And so Rosalie has been actually working for now quite a while to try to make these proteins inside here accessible. And what we have been using is actually something that's used commonly with a cell, which is basically antigen retrieval. So normally when you do fixed cell cultures, people would have an antigen retrieval step, which is by cutting these cross-links, either by heat or by various acid treatments. And so we then optimize this process here in the context of EV context, and basically using a heat-induced epitope retrieval so that we could then have a proper binding of the antibodies inside of these EVs. And so this is the same process that we had for the antibody microarray is kind of preserved, except we have an extra step here. See, I'm showing all the steps, an extra step that would allow us to retrieve this. And this was optimized, and PBS at 90 degrees is the one that gave the best results in our hands. So some people use urea, but here we use this quite straightforward process, and that allows us to then extract these proteins. And these are some examples now, some primary examples where we look at integrin beta-1, so something that's on the outside, and ALX1, which is expressed only on the inside of EVs. And we have also a number of controls where we don't permeabilize to really verify that they're not being expressed. But then here we can co-measure both integrin beta-1 and ALX1, so you have the blue and the red bars on the same EV sample by having an antigen retrieval process. And so these are, and again, using different capture targets, we see different expression levels. And this is then combinations of integrin beta-1 on ALX and beta-1 on HSP70, and showing that for both of these cases, and I'm not going more into the details, that we see different expression profile patterns in all different kind of conditions. And so this was good and worked well, but as usually, so this took several years, and I'm showing a few and not all the details because it would be maybe a bit boring. But one of the challenges was, for example, we had weak fluorescent signals, and so Rosalie, then inspired by our work from DNA exchange imaging, we then used oligoamplification inside of these EVs to try to actually rescue the permeabilized proteins, and so that we could also detect them simultaneously into these EVs. And this is an example where we have now different linear 2-branch here and 4-branch detection with the different signals. And as you can see in our hands, 2-branch signal was the one that gives us kind of both an adequate signal for intravesicular protein, as well as for extravesicular protein on the outside of the EV. And here is then a proof of concept heat map where now we could start measuring a number of intravesicular proteins together with extravesicular protein, and also potentially measuring simultaneously intravesicular, so the ectodomain of a protein as well as the extravesicular domain of the same protein to verify, for example, phosphorylation or different mutations that might, or cleavages that might happen on a particular protein. And this, so this is something that we'd like them to explore further in the context of the colorectal cancer metastasis samples, both for cell culture and for liquid biopsy applications. And so, coming to the one of the big challenges. So I spoke about single EVs, but so far I've only spoken about population analysis. And one of the challenges of population analysis is that we cannot differentiate between this type of EVs, for example, where statistically we'd have a fairly average expression of this brown protein here, to this situation where we have one EV with very little or maybe no expression of this protein, and some EVs with a very high expression of this protein. Because also these are not the same when we do an average fluorescent measurements, including with our normalization efforts. What we would see is the same fluorescent signal here for CFDSE, the universal labeling. But if we look at EGFR, assuming this would be EGFR here, we would see the same signal in both cases, but these are biologically very different signals and very different associations that we're having here. And so average signal really basically masks the individual single EV differences or small EV differences. The percentage of EV that express a protein is masked as well. And as we're seeing from CD81 or CD9, there's lots of EVs that don't express a particular protein, but we can't really see that very easily when we're doing these population type measurement. And finally, co-expression basically is lost. And so this is what motivated us to do a big effort on single EV analysis. And this is something that started with a nanoarray platform that we had developed. And that led to early successes. And the idea was that the nanodots would be small enough so that only a single EV would be able to be captured on one dot. That would provide us a size exclusion opportunity for capturing EVs. However, because of the printing and the nanodots are actually not, as we can make nanodots reliably at 50 nanometer, at least not in a mass production way. This was more a way of actually colocalizing individual EVs on the nanodots, which was running counter to actually our initial purpose. And so we kind of set that a bit aside, but it allowed us to demonstrate at least that we could have single EV detection here on these different nanodots, and single fluorophore detection, and with AFM overlay also confirming that indeed we could have single EVs at low concentrations and we will capture them. However, as Nusha was saying before, we had only 3 colors here, and when we now look at EVs, we would really consider that many don't express different proteins. We were very interested to having the ability to high multiplex analysis of EVs. And this is how we started basically then the DNA exchange imaging project several years ago, using the flow cells that we showed before. And one advantages of EV is that we don't have much DNA inside, so that's not something we have to be worried about, that the DNA would bind to the nucleus. But on the other hand, the signals of EVs are very weak, so the challenge here is then to detect the single EV binding. And so Philippe is the one who started this project. And one of the first challenges also was to find actually DNA barcodes that would allow us to hybridize. So Michelle was asking about controls. So one of the controls to make sure is that when you have one barcode, it doesn't cross-react with other. And so we selected actually large number, 96 barcodes here computationally. But then experimentally with microarray, we could see that a fair number of them would cross-react, which you see with these low signals. But nonetheless, from this collection, we could then tune selection of DNA barcodes. And here you have the scale bar where you have very little cross-reaction between all of them for the DNA exchange imaging setup then. And so as we saw with cells here, we would have a starting surface, which is shown here. Then we have a first labeling, for example, CD63. We would identify here small fluorescent spots, which can then, after displacement and wash, disappear, confirming that we have an efficient removal, which then can be followed by CD9 displacement here, that can then label the second label. And this can then be repeated, and of course, by using several fluorophores, we could then measure multiple colors at once. And this is one example of EVs from HT29 cells, where here we have 2 channel measurements, and we measured an integrin alpha 6 and integrin beta 4 in the first set. Then we measure EpCAM and ecadherin. And I don't have the highlights here, unfortunately, but there's actually a number of EVs that actually show up. I think these 2 here in particular are shown also here, and I think they're also shown here. But for example, this one is only showing EpCAM, but or the EpCAM signal doesn't show up here. So we see EVs that express 1, 2, 3, or almost none. So ecadherin here is almost not expressed, and we can now start to map basically these different EVs. And so far we have run 20plex to date, and so we're really coming actually to a stage where we can run this much more reliably and start to have this network maps of EV co-expression patterns. This is our library of conjugated antibodies to this point. We're looking to expand it, so if you have suggestions, we'd be happy to discuss. We have some ideas on our own, but of course, there's also new applications that we could consider. And so since I'm running short on time, I will indeed skip the whole part on the protein binding and just run to my conclusion here. And so I hope that I showed you that I've got some excitement for the new technologies for advancing science and investigating human health. So we showed you single-cell immunity affinity proteomics, and so we're really interested to characterize CTCs and to use that actually in the context of colorectal cancer, liver metastasis, to identify subtypes of disease. Because currently there's 2 subtypes. I mean, I'm simplifying here a bit, and they both receive a therapy, but for one subtype, the therapy is actually harmful. And right now they're giving it to everyone because they can't define the subtypes before the surgery. And so everyone is receiving basically a neoadjuvant harmful antibody therapy, which is also very expensive. And so we hope that with the CTCs, we could then identify that earlier so that we could actually save these patients from the therapy, which is for people into cancer. It's a bit like Oncotype DX, which can basically identify people who do not benefit from therapy, which is an immediate savings to both the patient and the healthcare system. I didn't show you the multiplex protein analysis. I mean, something that we have worked for many years, but that's now been spun off in a company, NPlex Biosciences. So we're actually having less activity right now, but we hope to start this again as we're ramping up. And then I showed you even protein analysis, where you have worked a lot on normalization. We have co-enrichment, intravesicular analysis, so really kind of a whole platform for measuring it both at the population level and then the single EV analyzer. And so we're excited to kind of moving out of the technology development really into the application. And so the application, so colorectal liver metastasis is one of them. We're interested to analyze CTCs and also EVs here, as well as cancer on a chips and organoids. And this is, of course, could also tie into stem cell models and so forth. And so with that, I'd like to thank everyone for your attention. And I'm sorry it was a little bit a long tour here, but I'm happy to stick around for questions for people who have time. And so thanks. Yeah.